could mute. Okay.
Yes.
Well, good morning, good afternoon, good evening, wherever you are. It is it's a great opportunity the COVID has given us to connect with people. And in this series of lectures, we thought we would introduce and discuss various aspects around cryo-electron microscopy and related techniques. Obviously, it has been an intense period of activity with cryo-electron microscopy itself around COVID. But then the technique itself has attracted tremendous attention because of its power of unraveling matter and the diverse details of matter and various phenomena. Today, we have an eminent practitioner of this technique. And I have heard her at least 4 times, I recall. The last one was the Gordon Research Conference in February this year in Texas, Galveston. Now, I saw her all these years as a person who is passionate about what she studies, that in those talks, they were all about atomically precise clusters. And our group, of course, has been working in this area. It's very nice to see a person who has been passionate about PMBA and looking at 10 ... or 68 or other cluster systems. The way they are, the passion has taken Maia to greater heights. I was so happy when this paper came out. I was seeing it. I was sort of excited about this paper in Science that is mentioned here, the first paper. And that was an eye-opener to many, what this technique can do. Of course, you will hear more about this. I would like Amrita to introduce this person of passion. Amrita.
Hello, everyone. I hope you have enjoyed yesterday's talk delivered by eminent physicist, Professor Miguel Jose Yacaman. In case you haven't, you could not attend, or any of your friends wanted to attend but could not, please follow our Facebook page, that is Pradeep Research Group. And I have shared the YouTube link also in the chat box. There also you can check. And today's lecture and the subsequent lectures will also be uploaded. So today is the second day of the lecture series on Cryo-EM and related techniques. And we have as a speaker today, Dr. Maia Azubel from Stanford University School of Medicine, USA. I'm personally happy to introduce Dr. Azubel because I am ever amused by the massive complex machinery of the advanced transmission electron microscopes. And therefore, the way Dr. Azubel has made this giant work for her is really inspiring. And I think she's a myth buster of the idea that women are not best suited for the heavy advanced machinery. So coming to her background, Dr. Azubel obtained her PhD at the Department of Genetics, Hebrew University of Jerusalem in 2005. And then she moved to Roger Kornberg's lab in Stanford, where she is a senior scientist. The main focus of her work was structural determination of big macromolecular assemblies. Now, atomically precise nanoclusters show physical and chemical properties which are very unique and very different from other nanomaterials. So therefore, it has gained a lot of attention from the material scientists lately. So Dr. Azubel has been the pioneer to use electron microscopy in the study of gold nanoclusters at atomic resolution. She has received many prizes and awards for her excellent contribution to science, like Menashe Marcus Award 2005 for excellent graduate students in genetics at the Hebrew University, Lev Margulis Award 2004 of the Israeli Society for Microscopy, and so on. Let me stop here and request Dr. Maia Azubel to speak on structures of atomically precise clusters by aberration corrected TEM and single particle reconstruction. I'll stop share now.
Okay. Well, thank you very much for such kind introduction. I'm honored to be here. I'm also inspired about the creativity that the scientific community is showing in order to keep us connected. And I especially want to thank everybody of you that are in completely different time zones, and for some of you it's quite late. So I hope that I will make this entertaining enough so everybody keeps awake for a little bit longer. I was mentioning in the introduction, I am really passionate about this work. And I have to say that, and especially, I don't know, but I'm assuming that there are students among the listeners and postdocs.
Yes, most of them are students.
Excellent. So there are some things from the science that I hope you will take from this talk, but also I hope that you will take a little bit from the journey that I did to get to the structure of gold nanoparticles, because the truth is that for a very long time, every time I was working with the structure of the gold nanoparticles, I was saying that's my secondary project. I'm a member of Roger's lab, and we are a lab of structural biology. We have different techniques that you can use when you want to solve the structure of different, let's call it, biological problems. And I'm not going to talk of any of the projects that I'm working that are related to structural biology today, but I saw that in December you will have a talk from Professor Wachu. Is that correct?
Yes, yes.
Okay, I strongly recommend that everybody goes. The work that his group is doing is simply amazing. But the truth is that with the introduction of the new cameras and the new microscope, but especially the new cameras, there has been a complete revolution of what transmission electron microscopy can do for biological samples. But before that, in Roger's lab, there were a lot of questions that need to be asked. And the reason why we got to work with gold nanoparticles is because there was a very clear motivation about that. That's before my time in Roger's lab. And there was the issue that when you solve a structure with X-ray crystallography, you have a structure. When you solve a structure with electron microscopy, you have a blob. So people from the X-ray field were kind of mocking the electron microscopies that we don't do structural biology, we do blobology. And the people from the electron microscopy were saying, okay, this is because you have the phase envy. Because the truth is that when you image, and we will get a little bit to what is that we are imaging, or what is that we see when we image, you get the phase information as well when you work with electron microscopy. So how we get from this motivation that we want to improve the resolution that you get with electron microscopy to work with gold nanoparticles. And that's how the heavy-EM proposal was born. And the idea was that we need to find an object that can be easily imaged under the transmission electron microscope when you collect the images close to focus, so you don't lose the high resolution information. And for that, you can use heavy atom clusters. We end up working with gold nanoparticles because the gold is a very good metal to combine with biological samples. But the idea is that instead of having all this protein that hardly scatter any of the electrons because it's composed mainly from carbon, hydrogen, oxygen, once in a while you have a phosphorus. So you will attach something that scatter electron very strongly. But in order to do that, you need to get a particle that is very homogeneous and you can attach it very specifically and rigidly. Otherwise, and we are going to talk about that in a little bit, when you try to do the alignment, you will have a problem if you have a lot of flexibility. So that's how the Kornberg lab, about 20 years ago, or probably more now, started working with gold nanoparticles. When I joined the lab, a couple of months after I joined the lab, Paolo Judzinski, who was a PhD student in the lab, was able to crystallize what ended up being the first cluster that was solved to atomic resolution by X-ray crystallography. And this is the gold 102. It was a very exciting time. Paolo was so happy when they got the diffraction pattern that I told him that he had to bring a cake for the lab. He didn't do at that time, but when the paper was published, he finally brought the cake and we celebrated. So I'm a new postdoc in the lab, and let me just go one step back and mention a brief comment about how you synthesize these particles. So you have a salt of gold, you have a thiol, and I have to correct a little bit from the introduction, and you will see in a second, my passion is for the 3-MBA and not the PMBA, and I will see why. But essentially, most of the thiol will work. And then the first step of this synthesis, you form a polymer, and then you take your polymer, you add sodium borohydride, and you get your cluster. So most of the clusters that were synthesized at the time when I joined the lab were clusters that were organosoluble. And because we wanted to mix them with proteins, we had to work with thiol that were water soluble. There are a lot of properties that are related to the core of the particle, but you will see that when you prepare these particles with the aim to bind them to protein, whichever is the thiol that you use is very important. So again, I'm a brand new postdoc in the lab of Roger Kornberg, where all of a sudden doing crystallography of gold nanoparticles seems very interesting, but we have a little bit of a problem with gold 102. The problem with gold 102 is that the synthesis was not very reproducible, and when we tried to attach to protein, it worked, but we wasted a lot of gold. So I was working in two fronts. The first one is, how do I get a synthesis that is very reproducible? And the first thing that I learned about that was that, as I mentioned, right, there are the two steps. If after this first step, I don't let the solution equilibrate and I add the sodium borohydride, I get, as you can see here in the gel on the left, a lot of bands. This is, again, the advantage of working with a water-soluble cluster is that you can run it on a page gel and you can see the different clusters. There are discrete size, but yet there are quite a lot of them. However, if I do an equilibration of the solution, then I get a very homogeneous product. And as you can see, I put the dates here just to mention 2 points. The first one is that the synthesis is reproducible. I was able to make 5 years later or more, 5.5 years later, the same synthesis, and I got the same product. But also to mention that this product is very stable. The sample that I made in May of 2011 was in my fridge, and when I ran the gel, it looked very good. The other thing that is important to mention about this synthesis is that sometimes everything works well when you are in a small scale, but then when you try to scale up, because you try to get a lot of clusters, everything goes down. Fortunately, when you do this trick of the equilibration, the synthesis is very stable and very reproducible for different scales. So now to the question of which thiol. And I tried quite a lot of them. again, doing the same trick, I was able to get this homogeneous product and a single band. And also I learned that if I change the ratio of the thiol to the gold, right, at the moment of the synthesis, I will get size of different, product of different size. Why this is important? Well, I always say that size is for electron microscopy, what color is for light microscopy. If we have different size, we can label different things simultaneously. And that's very important because, again, as I said, we were targeting all of these products to label protein and put it in the microscope. So that was great. Okay, so I'm there. I have something that is very pure. I'm in Roger Kornberg lab. Everybody is crystallizing things. They solve the cluster. So I want to do the same. So, oh, sorry. Before that, the reason why I like the 3-MBA has to do with the reactivity, right? What you're seeing here is a gel where I have a 3-MBA cluster and I change the ligand for glutathione, DTNB, the 4-mercaptobenzoic acid, or the N-acetyl L-cysteine, and then I incubate these gold nanoparticles with a small antibody that has a surface exposed cysteine. So I have a free thiol on the cysteine. As hopefully you can see, when I have the 3-MBA, the reaction gives a more predominant band if you compare to all the other ones that are faint. Where you have in the bottom is just the gold nanoparticle. And I tried that also with DNA. This experiment is a little bit more complicated because what I do is I have a cluster that is hybrid and I have a little bit of MBA on the surface and a little bit of glutathione. And what you see in the diagram here to the left is the first one is the free gold. The second one is when you have gold attached to one molecule of DNA that has a free SH, 2 and 3. And hopefully you can see that as I increase the amount of glutathione, the number of molecules of DNA that can enter the cluster or can be conjugate to the cluster is reduced. So the reason why I like the 3-MBA is also because it turned to be a great collaboration with Hannu Hakkinen and Mika Pettersson in Jyväskylä that has a lot of properties that I'm not going to be able to get into detail today. But again, as I said, I was there. And all of a sudden, this is my first few months, and I got crystals. And you hear all these stories about people that are very, very successful in the very few months of their postdocs. And I really struggled during my PhD to get the results. So I thought, okay, maybe that's me. Maybe all these stories that people are telling are right. And I got really excited, and then I was completely crushed. And I have the first and most important lesson that I got from crystallography. It's not that you're looking for crystals. You're looking for crystals that diffract. And my crystals never diffract. So essentially, I have all these beautiful products, and I'm starting to think that maybe I will never know the structure of those because I try all the different combinations. You have no idea how many trays I try to set up with crystals for that. And just as a reminder, this was my secondary project. But then the material science department at Stanford purchased a Titan, not a Titan Crius, but a Titan. And the images that you are seeing here were coming from that microscope. Those are images of silver nanoparticles. And I was especially interested in this small guy. This small guy was about the size of my gold nanoparticles. So the idea is, okay, now we have an electron that, an electron microscope that can image the particles to this resolution. And why is that this microscope can do it? Well, the trick is on this box here that we have in red that is performing the aberration correction. Why microscopes need this corrector? And that has to do with the properties. When you have a beam of electron that is crossing the lenses closer to the edge and not to the middle, those one, after they cross, they are deflected more pronounced. So instead of having a focal plane where all the information converge to a single point, which give you good resolution, you have this fuzzy image. And this is part of the issues that you have, and you will have also in a light microscope. It's just that you're working at different resolution and has to do with the point that the lenses are not perfect. So the aberration corrector, what it does is, in a very clever way, insert different elements that make the beam change direction in a way that when they converge, they will converge in a single point. And what are the advantages of the aberration corrected microscope? As is listed in this slide, reduce the localization, improve the resolution, and the fact that you have a negative CS means that each white dot that you see in your image is an actual atom. And I think that it's always better to show that with an example. So here you have the uncorrected on the left, you have the uncorrected image and the corrected image. And I believe those were Titan clusters. So the one thing that you can immediately see is that these white spots that we see on the particle itself, they are delocalized to what is the background, where here it's clear that the background is an amorphous surface and you have very clear details and you can see the edge of your particle very clearly. And that's what the aberration correction does for us. So great. Before we get into how I solve the structure, this is a very important point. And this is something that anyone who started working with transmission electron microscopy has to know. And it's the fact that what you get is a 2D projection of a 3D image. Oh, it's not working. Wait. Sorry. No. Okay, let's see. No, it's not working. Okay. That was supposed to show you that you can be fooled by thinking that this is actually a bunny, a shirt, a guitar, or a shoe, because actually, if we were to see that, we will see that they're actually, I'm worried now that the rest of my videos are not going to work.
So we can probably get out and then play it from the...
Okay, I'm trying to get out. It doesn't, okay. Sorry about that. Okay, let's see. Sorry, my computer has frozen a little bit.
All right, no issue.
Okay. Huh. No, okay, that video doesn't want to play. Let's move on. But it's important to know that what you get is the 2D projection of the 3D object, right? So if what you get is the 2D projection of the 3D object, the question is, how do you go from the 2D projection to get the 3D structure? And this hopefully does work. Yes. This is a video that will walk you through the entire process when you work with Cryo-EM. You apply your sample to the grid, and then you have to remove the excess water because you want a very thin layer where of solution where your particle is embedded, because then you are going to get and plunge freeze it into liquid ethane, and you want the ice to be amorphous. You want beatified ice. If you have crystalline ice, it's going to give you spots. Then you load the grid into the cassette, and you can, in the Titan Cryos, load up to 12. You put it in the capsule doesn't float. You have to take it with your hand, but that's the only thing that you have to do it manually. And when you have the grid in the microscope and you take an image, what you are going to have is, if you're lucky, different orientation of the particle. And then each orientation is going to be combined to get the 3D structure, okay? So this is what it's called single particle. In single particle, you take one image, you are relying on having all the different orientations. And then the other option that we have is to do electron tomography. Again, if we are doing cryo, the first step is the same. You put on the grid, you blot to remove the excess, you have a very thin layer, you plunge, but then instead of taking only one image, you take multiple images. Oh, this one is not going to work either. Oh, so sorry about that. No, something with the Zoom, it doesn't. Okay, in any case, what's going to happen, and if you can look at my hand, let me see if I can make it play from here. Can you see my screen?
Yes, we can.
Okay, and this is not playing anyway. Okay. Again, what you are doing, if this is your sample, the sample, you can start tilted or you can start it at 0, but let's start tilted because that's what you're seeing from, oh, there you go. So the samples start to tilt and you collect the different views. The problem you have here when you are doing the electron tomography is that the beam of electron is passing through your sample multiple times, okay? And that was a problem and has to do with the fact that these particles are very, very sensitive to beam damage. And hopefully this one will run. Oh no.
You may get out and then play.
Yeah, that's what I'm trying to do. So if I get this video to play, you will see how these particles, okay, loading, some reason, no, it's, well, what you will see is the particles start to merge, okay? So the problem is that, oh, here, they move very slowly.
Is it that there are many other programs running on your computer?
Could be. Let me close view. This. Oh, this I cannot. Okay. Hopefully they will start. Oh, so sorry about that. just close this too. They are not even closing the program, so let me stop the share for a second.
You can restart the PowerPoint.
Yeah. Mm-hmm. Close the programs now, please. Okay. Well, I will keep talking. Hopefully the PowerPoint is going to, I'm closing PowerPoint, but it's taking a while.
So shall we take a few questions?
Sure, if there are questions so far. Are there any questions so far?
I don't see any, but in case there are, people can ask. You are passionate about 3-MBA. So maybe let's talk about your passion of 3-MBA versus 4-MBA.
So the thing that we learn about the 3-MBA is that it's more reactive than the 4-MBA. The 3-MBA, I have 3 clusters with the 3-MBA, and I haven't had any issue to label any protein that has a surface exposed cysteine with the 3-MBA, whether with a 4-MBA, oh, now it's working. Okay, let's see. Okay, this is the video that I wanted to show first, where you cannot be fooled about what's happening with the 2D projections. Okay, and let me show you one more time. Maybe now will work like this as well. Yes. Okay, this was the point that I was trying to make, and I think that it's a very cute work that the people at Seoul did. So this was one of the videos that I couldn't show, and here is the other one that shows what's happening when you are taking the TILT series. And finally, hopefully this will work and you will see, yes, this is what happened when you open the beam and you let these particles stay there. Let me show you one more time. The beam completely melt these clusters. And what happened is that you deprotect the monolayer and they start collapsing to form things. And this is a problem because the structure that I want to solve is the structure that I have in solution, not the one that form under the beam. So for that, let me skip that. What I had to do is to use what it's called the low dose strategy. And this is very important because this is something that I took from structural biology and I applied to material science. The material scientists that were working with me were not very happy because their microscope was not set up. But this is the one thing that I mentioned to postdoc and students. If you know something from a different field that you can apply to a new field, just go for it because sometimes it makes all the difference. What is this low dose? Well, you search at very low MAC so you don't damage your sample with the beam. You select where you're going to take the image, but you don't take the image there yet. First of all, you focus on 2 spots that are close but not overlap with the area that you want to take the image. And then you extrapolate the value of the focus, and only then you move the beam while it's still blocked, or you move the stage, depending how you are working. And by the time that you take the exposure, that's the only time that your particles saw the beam. So you can control that. And what I did is I reduced the expose of particle during imaging that normally was done in material science, in some cases in a factor of 1 million, especially for those people that were working with tomography. So what I'm going to tell you now is I solved the structure of 2 of my 3 MBA clusters that I like so much because I can attach them to protein very easily. The first one is the small guy that I used to call it 3 MBA2, it ended up being the gold 68. And the other one is the largest that ended up being 144. Again, these 3 MBA2, I show you the crystals before, but they didn't diffract. And this is how the image look of the 3 MBA particles with 68 atoms. And I don't know if you can clearly see the white dots that are the atoms of these clusters that I image with the aberration corrected microscope, but that's why you do averaging. And why is that you do averaging? The reason why you do averaging is because your noise is random. So if you have a very noisy image, sometimes you don't see the details. But the moment that you start adding more and more, the picture becomes clear. And that's the reason why you do averaging in order to improve your signal to noise ratio. However, in order to do averaging that works, the alignment has to be good. Because if you have 2 particles that are exactly the same, not that noisy in that case, but you are going to average those 2, you're not going to get more information, you're going to get wrong information. And even though the software has changed quite a lot since 2015, I believe that was when or 14, when we published the structure of gold 68, the same principles apply, okay? So here again, we have the raw images. Hopefully now that I'm showing you the averages, you can see a little bit better the atoms because the noise of the background gets canceled and you increase your signal to noise ratio. And those are the classes that we use to produce the 3D structure. When I took the 3D structure and I took a cross section and I compute the Fourier transform, it was very nice to see that there were spots to 2.4 angstrom. So yes, this was the structure of a gold nanoparticle, so atomic resolution with an aberrated corrected microscope. One thing that is very important is to see whether or not all the steps that you're taking make sense. So what you are seeing here on the left is the distribution of all these classes that were used to build the 3D structure. And it's very important, each one of these bars, I will tell you exactly what it is, but it's very important that you see that you have coverage of all of this space, because otherwise what is going to be happening is that you will see that there are some part of your particle that has not been imaged, so you don't have the information that correspond to that view. So again, I already kind of told you, each one of these points is one view, and that view has like a projection that is associated. In order to check at that time that what we were doing makes sense, we have to inspect that the projection, that is the first column that you see here on your left, on your right, the projection matches the average, that is the middle column here, and that the individual particles that you have on the farmost right are consistent with the class and the average. So when we saw that, that was a very good indication of the robustness of the reconstruction. The map, however, didn't look very symmetric. I was a little bit disappointed. And again, unfortunately, I will not have time, but please, for people that are interested in cluster and 3 MBA, go and find the Tiarica Taro paper, and it's an AS Nano, where they explain the effect that the 3 MBA this cluster. Sorry? Oh. Okay, so one of the things that I did with this cluster is do a peak search. And that got more interesting. When I did a peak search, I found 68 peaks. Even more interesting than that was the fact that the distance between peaks was between 2.7 and 3 angstrom. And we already knew from the goal 102 that was solved by X-ray crystallography and other cluster that came after that that was the distance that was expected. So yes, each of these peaks that I was seeing were the peaks that correspond to gold atoms. The fact that it was 68 was very encouraging because before I got access to the microscope, we have done a lot of things with this cluster, including mass spectroscopy that was done by Professor Tsukuda in Japan. And we knew that we were dealing with a cluster with 68 atoms of gold. So after the structure was solved, we did a lot of validation. Part of the validation was done at Berkeley. Greg Hura did the SACs. Hannu Hakkinen and his group did the DFT. Mika Peterson and his group did the infrared and the UV spectroscopy. And it was very nice to see what you are seeing here. In orange are the coordinates from the EM, and in yellow, the coordinates from the DFT, that we have the validation of these things. So about the symmetry, it's not completely true that the cluster has no symmetry at all. The center of the cluster is a cube octahedron with an atom in the middle. There are about half of the atoms that extended into what is an FCC-like symmetry, and then the most exposed gold atoms are quite deformed. And again, that has to do with the fact, and I will say it briefly because I think that it's fascinating, with the 3 MBA instead of the 4 MBA, you have the carboxylic group that is in a meta position, and that carboxylic group can interact with the core. And that it's part of the suggestion that Mika and Hannu are making for why these 3 MBA deform the very symmetrical clusters that you can see in other cases. Luckily, there was another cluster that was solved at that time or a little bit before us that also have an FCC-like symmetry. So we knew that there were a lot of things that were together for us. And the other cluster that I mentioned is what I call the 3 MBA 7, and it turned out to be the 144, but not the 144 that people were working with organic thiols or with the PMBA. This is again slightly different because of the use of the 3 MBA. But what you are seeing here are 2 images, one of the F20. The F20 is the one that doesn't have the correction, and in that case, you will not be able to see the individual atoms. The magnification also, it's much lower, but you can see how nice and homogeneous they are. Also, another thing that it's very important for the application of the single particles is that you have monodispersed particles, and that's what you can see here on the F20 image, but also on the Titan. The Titan is the one that is averaged and corrected, and I don't know if you are going to be able to see how gorgeous this micrograph is and that you can see the individual atoms, but this was a sample that was really nice to work with. So when I saw the map, it looked like this. I did the peak search. So I think I said, oh, here, you got the symmetry you wanted, right? We like symmetry. But again, remember what I show you finally when the video work. Don't get anything fool you just by seeing a 2D projection of the 3D object. It's true that most of these atoms are organized in an FCC kind of symmetry, but if you see, there are some that I indicated at a small sphere, and those completely deviate from that symmetry. So a more honest picture of the cluster is this one, okay? This is how the core of my Goal 144 look. And this is something that I didn't mention, and it's the fact that unlike crystallography, with EM, we can only see the core. We don't see the ligand, and that has to do with the fact that probably by the time we finish imaging, the ligands could be destroyed, but mainly because the ligand has atoms that are so much lighter than the goal that we don't get that signal. So one brief comment is that by the time I solved the structure, there was a lot that has been mentioned about the optical gap of the clusters, that most of them were superatoms. There was a clear tendency, and my two clusters fall off that curve, which means that for the same size, were much better conducted than predicted. And again, this is something that has to do with the lack of symmetry. And that's something that you can see on Hanu's paper. When you have lack of symmetry, the molecular orbital relax. So the gap that you have between the HOMO and the LUMO is smaller. So I apologize for all the technical difficulties. I think that I was able to mention a little bit about the synthesis and what was the key for me. I didn't say much about the 3-MBA other than the fact that actually summarized is a cluster that it's very reactive. So I can attach it to any protein that has a free thiol, any nucleic acid that has an SH group. I haven't found one part, and I'm telling you, after I gave a couple of talks, I got particles, I got protein from a lot of people say, oh, could you label this one? Could you label the other one? And with the 3-MBA, always work. The 3-MBA can be for ligands. So after I form my conjugate, is inert, and I use in the other of the project. And I mentioned this thing of the optical gap because I really like the fact that the structure allow us to understand a property that we didn't know how to explain before. How was possible that a cluster with 68 atoms was conducting much better than what the prediction was doing? And that has to do with the lack of symmetry. But I hope that the most important thing that you take today, even though I didn't talk about cryo-electron microscopy, cryo-electron microscopy has also the advantage of working together with your sample in order to protect it from beam damage. What do I mean? I mean, if you are working in the cryo condition, by the time you damage your sample, the ice is already melt. So you know how to work in that case. And also, as I mentioned, is that one of the reasons why this project was successful was because I took a technique from a completely different field, from structural biology, the low dose, and I applied that to the material science. With that, I have a lot of people to thank because this was an incredibly collaborative project, and I think that that's probably one of the goals that you have with your new center, to make it a collaborative effort, and I think that that's the way to do science. So we have expertise, and I mentioned Hanu and Mika, and I mentioned Professor Tsukuda. I didn't mention Eileen. She was the one person working with me for the microscopy. Everybody in the Kornberg lab, especially Dave, he was a lot of help with a lot of the computational things. And I can never thank enough Roger, who is an incredible person to work with, is super supportive. Apologize again for the technical difficulties, but I wanted to leave sometimes if there are any questions.
Thank you very much. It was highly illuminating lecture, and your passion came out despite all the the minor troubles with technical issues. We have some questions, and so I would like people to ask, write the questions down. At the same time, I would also allow, or I would like to give time to people to ask, articulate the questions the way they want to articulate. So let me ask Subbu to ask the first question. Subbu?
Thank you, Professor, and thank you very much for the wonderful and lucid presentation. It was like it was at both at a basic level and from the application standpoint, it was very interesting. So I have a question. So when we normally do structure analysis using single crystal X-ray diffraction, we kind of refine the structure. We put the structure to a lot of tests, and we refine it and kind of look at the R value or R square value, and then go back and work on it if the value is not good. So how does this process work in when we arrive at the structure using electron microscopy? That's the first part of the question. And just as a follow-up, does the structure, final structure, depend on the number of images that are being processed? Yeah, thank you.
Both questions excellent. Let me start with the second part of the question. Yeah. So, on that, I think that it's going to be good for me to give you the example. The reason why I published first goal 68 is not because the cluster is small. It's not because I collected that image faster, that data set first. It's because goal 68 has less tendency to fall in one side on the grid, okay? So in order to do the single particle, you need to have all the different orientations. So what happened was that when I collected and I showed this plot, when I was trying to reconstruct the goal 144, what I was seeing, and I was trying to look if I add the slide, but I see that I remove it. So what I was saying that instead of having these columns all over the space, I only had a halfway. So in that case, I had to go back to the microscope and collect again, because what was happening was that I only have one portion of my structure that was well represented and the other one was not. Normally what happened there is that when you see specific projection, you get a clear idea of what your cluster looked like, but in other projection, it's very difficult. So it's always true that when you collect more data, you get better. Well, there is a point that at a certain point, you don't need more particles. What is happening if you follow any of the work that has been done with structural biology of proteins is that it turned out that the proteins are quite flexible. And the reason why people keep collecting more and more and more and more is because what they are trying to do is to sort between different conformation. The truth is, for things that are very rigid, normally if you have enough information to cover all your orientation, you are going to reach a point that by adding more particles, you don't get more resolution. And the structure would not depend on the number of particles. The resolution sometimes does to a certain point. And that gives me to the point of the refinement. So again, most of the work that has been developed for the software of this single particle entomography has been done for proteins. So up to 5 years ago, it was a very, very difficult question to ask because we didn't have the resolution to build maps and refine and to see side chains and to see individual atoms. But in general, what happened with this processing is as similar as you do with crystallography, you go through multiple cycles of refinement. I just mentioned that as a simplification, but what you have is that you get your images, you get your classes, you form your 3D object, then you reproject the 3D object, you use this reprojection as new classes to classify your data set again, and you start all over again. Eventually, when you do this process, if the process is going on the right direction, it will converge. And the moment that you converge is when you stop. But you still need to check this. You still need to check that your projection, your class average, and your particles make sense. Otherwise, something went bad. Now, with the new microscopes and the new cameras for structural biology, there is new software that has been developed. People start talking about the V factor now with EM, but I think that that was not possible at the time that I solved the structure, so we don't have any R factors to report with these structures.
Thank you.
I hope that answered the question.
Yeah, thank you very much.
Well, we have several other questions. Let me ask people one after another, and as much as whatever time that we have, we'll take all the questions. So if you please stay back. And in case we don't get there, I will share these questions with Maia Azubel, and so that she can respond.
Be happy to.
Next person, Dr. Tripti Ahuja, you have a question?
Hello, ma'am. So I have a question regarding, you showed some HRTEM images of silver nanoparticles. So those were like excellent. I just want to know, is it like depending on the material and the type of ligand or the stability, so that we get these high quality images? Or is it like a universal kind of thing that it will be come with each kind of AGNPs, like silver nanoparticles?
So there are a couple of ways to answer. So the one reason why, especially let me just try to put it back to because I have the animation. So one of the reasons why, especially the larger clusters, right, they look so good, is that the amount of dose that they use to image them is very high. Okay? Yeah. So that's one of the reasons where you have like this really nice. You can see that also in this small guy, you see very clear atoms. So I was not involved in this project. I don't have the answers, but I can tell you from what I've seen in different is that if you work in conditions that you prepare, it's very important to prepare your grid to be very flat. You have to be very careful always when you work with EM with the flatness of the grid. But especially when you work with these clusters, if you want to image gold atoms. And the reason for that is because the height of the grid is going to have an effect on the focus that you make. In order to see atoms of gold, you have a window of a couple of nanometers that you can go under the focus. You cannot go more than that, okay? Because otherwise you don't see the atoms anymore. So if your grid is not very flat, what happens is that part of your grid might be in a slightly off position, so you will not be imaging that well. But as far as I know, the ligand doesn't have much of an effect. Again, those were selected from galleries, but in theory, you are able to image this quite nicely.
Very nice. Let's move on to the next one. Manoj, you have a question on ligands. Manoj. Well, the question is that, if ligands are destroyed, how do you know about the structure?
How do you know about the structure of the ligands, or how do you know about the structure of the core?
If the ligands are going away, of course, there can be changes in the overall core as well.
Correct. So the honest answer is, and we can say the same for any method, right? How do we know that when we crystallize, we are not changing, like the crystal constant don't change? So the truth is, there is a possibility that just by putting the clusters in the microscope, you are changing. The one thing that I did is I took images at a dose that I could take 2 consecutive images to make sure that nothing was changing. So if there is something that happened very, very fast, I cannot detect it. But I was trying to be as careful as possible to make sure that I'm working with the minimal dose possible, that I still see signal, but that I don't see a change in the overall structure.
Very nice. So Dr. Ammu Matthew?
Hello, thank you for the great talk. I enjoyed listening to you. I had a question, probably you might have touched upon this sometime during your talk, but have you tried any organic ligand stabilized clusters on using your cryo method? I was just curious how they would behave. I would expect some aggregation, but have you just tried any? Okay.
I haven't. So the reason why is, again, we are a structural biology lab. We want to have things that we can attach. We are not well equipped to do some of the structures, some of the procedures that require the organosoluble bed. Also, I'm not so concerned about aggregation of the clusters to the surface, but for sure you will have to change a little bit of the property of the surface, because the carbon grids that you use for the EM work really well with water. You charge that and the water spread and it's very nicely. If you're going to put an organic solvent, you have to make sure that that also spread. So it's not impossible, but it's not something that I have tried.
Okay, thank you.
Sayan Kundu.
Hello, ma'am. Okay, may I ask one question?
Go ahead, please ask.
Yes, ma'am. So far, I understood to some extent that aberration correction. Okay, so it's like so far I understood that white field to confocal conversions as like that means we want to remove the out of focus light, and we want to put a light on a single point, and that is going to be made by aberration correction. Am I right?
No, either spherical aberration is corrected. So she was showing spherical aberration, and that you can see that in her ray diagram. Yes. So she is correcting spherical aberration.
Exactly. So that is the light which is coming from the edge of the lens. That is coming from the edge of the lens that are going to be focused on a different point, and that we don't want. That's why we're trying to make all the light to focus on a single point.
Yes.
Yes. So that's yeah. So ma'am, in next, I just want to know that can you please explain once more the low dose problem what you said, and that is serving also to fulfill the same problem that I couldn't understand. Can you please?
Okay, so there are two. So I think I understand the question. There are two different things. So the sample is aberrated and corrected all the time. Okay, which means is that when my beam hits the low doses, when the beam hits the sample that I'm going to image, it's doing it only once, and that's where I'm using the low dose. But the beam that is used in the low dose is being corrected, okay? It's just that you don't need to focus on where you want to image. You can focus next to that, okay? And on to the other side, and then you focus on the two sides. And because what I was saying, the focus is related to the Z height. If you have these two values, you can extrapolate to that, okay? So the beam has been corrected for the problems that you have in the lens when you focus here and when you focus here. So that's one thing that the microscope does by changing the path of the electron beam. But then is when do you allow the electron beam to see the sample? And that's when you use the low dose. That answer your question?
Yes.
Very nice. And so we are coming to the end of the questions. And I would go to the last question, really not a question. In fact, one of the students has this question. May I ask Sujan to ask the question that you wanted to ask?
Yes, sir, definitely. Ma'am, it was indeed a very nice talk. I am a student just starting my PhD journey. So I am a bit interested to know, if you can share some of the event or any kind of interesting story you are having with Professor Kornberg. So it will be a great pleasure for us because, of course, he is a Nobel laureate and eminent of the field.
Sure, I'll be happy to. I have many stories. I've been with Roger for 14 years now, and for me, it's a pleasure. Roger is a very humble person. when I joined the lab only a couple of months before he was awarded with the Nobel laureate, and it was a very funny day. And I have to say, I probably was the only one that didn't know about the announcement, because I got to the lab and I saw all these balloons and flowers and cameramans, and I have no idea what was happening until I went and I checked my email and people were telling me what was happening. But Roger didn't want to celebrate. We wanted to celebrate because it was a huge achievement. We want to throw a big party for him, but he didn't let us. It was like, so we went and we celebrate by ourselves. But the funny story from that day that I think that was great, it was Roger came to the lab the day before and said, tomorrow I'm going to Pittsburgh. He was going there, he was getting another award, and all of a sudden he had to cancel his flight. So he told us that in the morning he called to the flight company to say, oh, I'm sorry, I need to cancel my flight. Can I get reimbursed? Or I don't know, I need to cancel my flight. And the person asked him, why are you canceling your flight? And Roger said, well, it's just, I was just informed that I won the Nobel Prize in chemistry and I need to stay at Stanford for the press conference. And the person on the other side of the line started laughing and saying, oh, this is the most creative excuse that I've ever heard, but no problem, we can reschedule. Like that person didn't know. So Roger is a person that is really, really good to talk to. He always encourages people and he believes in project. If you think that I'm passionate about data, you should see how he reacts when I show the new images that I am collecting in the microscope. And it's a really joy, and I'm very grateful to work in his group.
Very nice. I suppose, Sujan, you got enough insights.
Yes, of course, sir. Of course.
Thank you very much. Well, it has been a great journey, and it's the passion that you have been, keeping, and the fact that you have been enjoying every day working with 3MBA and other systems that you have been. But then there is a lot of pain as well. And collecting quality data would mean that there are 100 other days wherein good data wouldn't come. And it is also a tremendous pressure. Well, everyone is succeeding with crystals and structures, and nothing comes out here is also a... But then despite all that, this has gone on. Obviously, it has given immense motivation to a number of others to pursue along this direction.
Hi, Maia.
Yeah.
Hey
Hi, Maia. This is Lourdes Xavier. Thank you for accommodating me in your lab when I was there in Stanford. I have a question. Like you might have seen the recent paper from Christopher Russo. They're using golden grids, and they are like the sample movement is restricted to sub-angstrom resolution because they have really, really small holes, and the ice movement is restricted. And he extrapolates that we can achieve, in principle, 0 dose, right? So let's say that gold clusters, if they are identical and the ligands arranged on them, they are not immobile, they are not mobile. That means they are all like as if they are in a crystal, they are all the same. Then how many images if we acquire, then we can actually solve the ligand structure also with electron microscopy if we achieve 0 dose.
Well, I have to say I haven't seen the paper, and for the sake of time and respecting everybody, I would just give a short answer. If the ligands are rigid, I believe that with the new cameras, you should be able to resolve them. You will have to be careful about the fact that I mentioned that you are working with both heavy atoms and light atoms, but the potential is there.
Yep, thank you.
Well, thank you. What I was saying was that there is a lot more to do, and glad that Lourdes Xavier asked that question. There is much more to do in the context of ligands and the kind of things that you yourself are pursuing with proteins and conjugating them with DNA. Many things are to be learned. And obviously, it's a global collaboration. Many more opportunities are there to explore this science. On that note, let us close for today. And tomorrow we will have one more wonderful talk. Again, using, well, this is more towards biology tomorrow. And that is a slightly earlier time. It is tomorrow's talk is going to be 5:30 p.m. Indian time. See you tomorrow. All the very best to you. Thank you, Maia.
Bye. Thank you very much.
Thank you all.